Biology, Part 186. The tension thus created is transmitted back down to the stem into the roots, and water flows into the leaves along the transpiration stream. Experimental evidence for the cohesion tension theory has been obtained in a variety of ways. In one experiment, a device called a dendrograph was used to measure the diameter of the trees. It was found that during the day, diameter was less reflecting and reduced pressure, and in the xylem vessels heated in its progress up the stem. In another experiment, sap xylem tissue was heated and its progress up the stream followed using a thermocouple. It was discovered that the rate of transport varied with the time of day, and that rapid transport in small branches started in the morning before rapid transport in the main trunk. This result can only be explained if the upward movement is the result of the pull originating in the leaves. It is clear that the cohesion tension theory can hold only for the unbroken columns of water. A bubble anywhere in the xylem vessel prevents water transport. Breaks may possibly occur from time to time, but it seems likely that they can be repaired by dissolving gases in the cytoplasm of the surrounding cells. Control of water. The process of transpiration is very wasteful of the water. However, plants control the loss according to the external conditions and internal demands, and are therefore able to osmoregulate to a limited extent. This is achieved mainly to the opening and closing of the stomatal pores, and these stomatal pores, consequently, transpiration is linked to the control of the gas exchange, which also occurs through stomata. The stomata are usually closed at night and open during the day when photosynthesis is in progress. This is made possible by changes in the shape of gut cells, which depend in turn upon the changes in water potential. The most important factor involved are thought to be the availability of carbon dioxide and ATP. When the gut cells are exposed to light, the concentration of carbon dioxide falls and the ATP is manufactured by cyclic phosphorylation explained. These changes activate a K+ pumping mechanism in the cell membrane and gut cells, causing the uptake of the large numbers of K+ ions from the surrounding epidermal cells. The cell's water potential becomes more negative and the water is absorbed by osmosis, making the gut cells turgid. As the gut cell swells, it changes shape because of the cell's walls bordering the stoma are particularly thick and resistant to the stretching of the, while those on the other sides of the outer sides are thin and flexible. Consequently, the gut cells become crescent shaped and hold the stoma open. At night, carbon dioxide accumulates as a result of the respiration and supplies of ATP are used up. The K+ pump is inactivated and gut cells lose K+ ions and become flaccid. As a result, they collapse, thereby closing the stoma. Under drought conditions, the closing of stomata may involve abscisic acid, and the details of this are not fully understood. In desert plants, including cacti, stomata are open at night and closed during the day, so that different mechanisms are required. So that's in fact particular to the desert plants, not to all of the plants in fact. The root pressure. If a stem is cut just above the ground level, sap can be seen to exude from the cut surface under pressure. This pressure is called the root pressure and can be measured using apparatus illustrated. In the tomato plant, the root produces an upward force equivalent to an astonishing 8 atmospheres, but in most plants root pressure is found to be much less, no more than 2 atmospheres. In some plants, including pine trees, root pressure is absent.